We're now moving into the second scientific theme. And to kick this off, we've got two science talks to sort of get us thinking about some of the big science questions we might address under this theme. And René Boiteux, University of Minnesota, he is now. It's gonna kick us off with that. Take it away, René.
Great, thanks a lot, Al. So I'm René Boiteux. I recently moved from Oregon State University to the University of Minnesota, and my lab develops analytical approaches for quantifying distinct molecular components of organic matter. And so the composition of dissolved organic matter matters because different components engage in different processes, right? So we have labile organic molecules that are major conduits of energy, metals, and nutrients. We have refractory components that act as this major reservoir in the ocean. Some organic matter components can partition into particles, and this may represent a sinking flux that's hard to capture by sediment traps, things like that. And then one of my favorite aspects of it is some organic molecules will bind to metals very strongly and with a range of specificities, and these have important regulatory effects on the solubility and bioavailability of metals. And so not only does the composition matter, but specific molecules can matter a lot. And so on the side here, I have an example of two different molecules that have very similar spectroscopic techniques and both bind to iron with similar affinities, but they have totally different effects on iron solubility. So this compound here, enterobactin, is a siderophore that's produced to solubilize iron. It forms a one-to-one complex that keeps iron in solution. But this compound here, tannic acid, is a degradation product of organic matter, and it's sterically hindered. So tannic acid ends up forming these networks of molecules that are different molecules binding to a single iron that create a mesh that precipitates out of solution. So two different molecular structures end up giving us completely different behaviors. So it's important when we see organic ligands in the ocean to understand, are they going to enhance the solubility of the metal and affect bioavailability, or are they going to enhance colloid and particle formation? Molecules can also provide clues about the fate and precursors of organic matter. So this is a depth profile showing dissolved organic carbon concentrations from the Bermuda Atlantic time series. You have high concentrations at the surface and lower concentrations at depth. And many decades of work by Dennis Hansel and Craig Carlson and others have shown that in the surface, we have some fraction of organic carbon that is indicative of a semi-labile component that turns over on the order of weeks to months. Extending into the mesopelagic, there's some semi-refractory component we can infer that is turning over on the order of decades. And then throughout the ocean, there's a refractory component that's turning over on the order of thousands of years. And again, these are inferred components based on concentration measurements. We don't actually have any insight into the molecular or chemical differences between these groups. So it's really hard to understand why some are short-lived and why others are long-lived. So our goal is to develop some tools that allow us to get a molecular picture of the chemical forms or elemental speciation of organic matter. And so our goals are to identify the major forms and quantify their spatial and temporal variability. If we can do that, we can start to categorize them into different key groups. For example, we can differentiate biomolecules that are produced by microbes recently for some functions versus degraded materials that can accumulate as more refractory components. And then we can start to develop overarching rules that describe how the ocean functions. So, for example, get at elemental cycling rates. And so the two tools that my group works with are liquid chromatography coupled with different types of mass spectrometry. And so one of the first tools we use is inductively coupled plasma mass spectrometry. So this allows us to take an extract of organic matter from the ocean. So in all of these examples, we're dealing with just a component of the organic matter that we can extract, and we can do different extractions to get at different components. This is a solid phase extraction using polystyrene. We can take that complex mixture and start separating it with chromatography and then detect the metals and other elements associated with it with ICP-MS. And so we can see an example of a chromatogram here where as different elements, different molecules are eluting from the column. If they have, in this case, we're detecting iron associated with them, we get a signal. And the area under the curve is proportional to the amount of iron that is associated with different reservoirs. And so in this case, this is a sample collected from the California current system. We can see sharp resolved peaks. These are indicating specific biomolecules or specific molecules that are binding to metals. And then we see this broad chromatographic unresolved mixture a chromatographically unresolved mixture that's indicative of degradation products. And so we can start slicing up these chromatograms and quantify independently, instead of just bulk iron that's bound to these different forms. So this is a great technique for getting at absolute concentrations, but it doesn't tell us the identity of the molecules. And so to do that, we can take the same chromatography and couple it to ultra high resolution soft ionization mass spectrometry. And so now, instead of just detecting the elements associated with organic matter, we're detecting the intact metal complex, intact organic molecules. And so at every retention time, we get a spectra that has thousands and thousands of different molecules. So we've been working really hard on approaches for identifying the specific molecules that comprise these different peaks that we can detect. So we've been working, myself and my postdoc Christian Dewey have been working with Yuri Carrillo and William Q at the Pacific Northwest National Lab to develop data processing pipelines to assign molecular formula to these LC ultra high resolution mass spectrometry spectra. This is based in a data informatics platform called CoreMS being developed by PNNL. So what this allows us to do is assign molecular formula to most of the mass peaks that we've had here. And from that, we can figure out which ones have certain elements associated with them, like iron, right? So that these techniques allow us to identify the specific molecules that we're detecting here. So in this case, these blue peaks were identified as amphibactins. These are specific microbial siderophores that are produced by certain bacteria in response to iron deficiencies as a strategy for solubilizing more iron and facilitating its uptake. We also often detect things that we can determine molecular formula to, but they don't match any molecule that is known to be produced by marine organisms, right? So these are orphan molecules that we can start to map their distribution, but they don't currently have a known biological function. So this kind of matches what we know about marine microbial ecosystems, that there's still a lot of unexplored microbial diversity, right? And a lot of the functions are still unknown. So what we can start to do with these techniques is map out the distribution of these different metal forms and try to understand their different sources and sinks. So that's what we did on this cruise in 2020 along the Oregon coast. This was a cruise led by Jim Moffitt, where we sampled across the Newport hydrographic line going from the coast out to the California current. And our goal was to be able to understand what are the sources of these distinct iron species that we can start to detect. So my students, Nicole Coffey and Maria Alvarez, collected samples with me, and then Peter Bright analyzed the samples and then developed a lot of informatic tools to be able to efficiently go from the raw data to distributions that we can plot in ODV and compare against other oceanographic data. And so this plot here is showing, this bottom plot is showing the distribution of just these unresolved iron complexes. And we can see very clearly when you start plotting their distribution that we have significant sources from sediments, right? And then that's supplying water, supplying iron bound to these unresolved, they're basically degradation products to the deep ocean and then to the surface. And then in the surface, we have these amphiphilic siderophores, amphibactins, that only accumulate in these waters in the surface offshore, where we have very much lower inputs of these dissolved iron complexes. So these are high nitrate regions with low iron, so iron deficient waters, and these amphibactins are basically the strategy that microbes are using to assimilate more iron, fish for more iron in their environment in this iron-starved region. One of these unidentified metal complexes we were able to see is really highest abundance in these benthic systems right near certain sediments. And so again, this points to the fact that there are certain microbial processes that are likely generating soluble iron, and these might be important for liberating iron that ends up reaching overlying waters. So we can apply these techniques to a large variety of elements in the ocean. The diversity is really crazy and striking. So any element you can detect with LC-ICP-MS, we can at the same time collect a chromatogram for. And what we start to see is that there's this huge diversity of metabolites and also degradation products that associate with lots of different types of elements. Here we have first row transition metals, so iron, but also copper, zinc, nickel have this combination of biomolecules and degradation products that we can start to quantify and map out. Halogens, even rare earth elements, things like cerium, seem to have these components of degradation products that incorporate those elements and potentially even some biomolecules. And so LC-ICP-MS, again, is great because it gives us quantitation of these different pools, but it doesn't perfectly resolve everything, right? So this unresolved material, right, is these degradation products probably represent a mixture that might have distinct sources. And so we've been starting to investigate the use of LC with ultra-high resolution mass spectrometry to look at distributions of molecules in a way that actually resolves the molecules that you can resolve with these high resolution techniques. Okay, so here's an example of that type of analysis. So in this figure, we're seeing the concentration. Each column here represents a single metabolite feature, DOM metabolite feature. These are masses that we assign molecular formula to within some retention time window in our LC mass spec data. And then their abundance in all of our samples, which are ordered from depth. So we have shallower samples at the surface and deeper samples at the bottom. We have about 15,000 features that we're able to detect across all of our samples, and we have relative abundance for them now at different depths. So using some k-means clustering algorithms, we're able to group them into distinct groups based on their spatial distributions. And this figure floored me because when I saw it, it immediately reminded me of that figure of semi-labile, semi-refractory, and refractory organic matter, right? Which is basically what these different molecular components represent. And so now we have thousands of molecules that belong to each of these distinct classes. So what can we do with that? We can start to compare their molecular properties. These plots are showing the distribution of molecular properties across these distinct groups. So the refractory dissolved organic matter has higher hydrogen to carbon ratios than the semi-refractory or semi-labile components. They're smaller, and they also have earlier retention times, which means they're more water soluble. So more refractory DOM groups are basically these more water soluble molecules. We can also look at whether these molecular features are corresponding to specific biomolecules or degradation products based on this dispersity index, which determines if something is a sharp chromatographic peak or a broad unresolved mixture. And we find that basically, even in the case of semi-labile DOM, most of the material that we're detecting is polydispersed, degradation products, and that's even more the case in these refractory components. So we can start to build out this picture, going back to the initial mystery of why do we have differences in reactivity of DOM. Our analysis sheds light on that, right? We have persistence of these smaller, more water-soluble molecules, and so it seems that you get this polydispersed degradation, generation of polydispersed degradation products on the surface that create more hydrophobic molecules that are removed more quickly, either by partitioning into particles or by being broken down by microbes because they're more hydrophobic and easier to stick to these aggregates. So to summarize, these LC-MS approaches enable us to resolve and quantify chemically distinct fractions of organic matter in ways that we can start to link to other environmental processes. And I talked about some examples how they shed light on iron cycling and on bulk dissolved organic matter persistence in the ocean. And so I'd like to acknowledge my group and colleagues at PNNL and on the two cruises. And I will take questions if I have time.
Thank you, Renee. We've got time we've got two people with their hands up, so let's run the mic to them. Ask your question. Mark, you can put your microphone on.
Jeff Bone from Scripps. That was a really great talk. So this is probably a profoundly naive question from a non-chemist, but are you able to treat those high-resolution masses the way that we treat like an amplicon sequence variant, where if you recognize that mass, that exact mass in one part of the ocean, and a different study recognizes it somewhere else, you kind of think it's the same thing, so you can build up a pattern of global distributions of these molecules? Yeah, so that's the assumption that we're making in this data set. And I think it's basically you're assuming that something at that retention time with that mass is that some molecule or something very, very similar to it. So basically, yes, we can start to use this to map out these different molecules and their distributions to get at sources and sinks and link them to things like amplicon sequences.
Okay, Ricardo, quick question.
Again, Renee, great talk. Thank you very much. Ricardo Letelier, now at NSF. What scares me about this is that it completely changes our concept of niche in the system. So the concept, the ecological concept of niche, when you have all these different molecules and all these different possibilities, each one of them in itself can correspond to a niche. So how do you think that in ecology itself?
Yeah, so I think, this is something I've thought about so much. It's basically the question of, you have this incredible complexity of organic matter, right, that somehow simplifies to things that have similar distributions that are functioning in a similar way. And so these clustering approaches are able to kind of do that, right? We're grouping things that are behaving similarly in the system to get at what are the molecules that have certain sources and certain sinks. Organic matter, these degradation products are probably generated through processes that take some larger molecule and then make lots and lots of changes that are somewhat stochastic. And so, like those processes, generate this complexity. But I think there are overarching rules that govern, like, okay, there are certain molecular properties like solubility that affect how these are going to continue to react. In terms of maybe your question is more along the lines of like how microbes deal with this stuff, and I think that
we're going to have to move on. Sorry, guys.
Yeah.
You've got plenty of time to get stuck into the niche and dissolved organic matter. Let's make sure we've got enough time. Thank you,
break
René.
it down piecemeal, is I think the answer.
Sorry, René. Okay, let's make sure we've got enough time also to hear from Mar Benavides from Marseille. She's going to talk about, I suspect, nitrogen fixation. Yeah. Particle-associated nitrogen fixation. Take it away.
Hi, everybody. Well, thank you to the organizers to invite me to give this talk. So yeah, I'm going to talk about nitrogen fixation, this time associated to particles. So just to give you some background, I guess everybody in the room knows, but nitrogen fixation is the reduction of a molecular nitrogen into ammonium. This is done by prokaryotes called diazotrophs. And this is a main, can you hear me? Yeah. This is a main entrance of reactive nitrogen into marine ecosystems, particularly in low nitrate regions such as subtropical gyres. So this fuels primary productivity and eventually organic matter export. So when we think about diazotrophs, usually tend to use this gene marker, the NIFH gene. And traditionally, we have based our studies a lot in cyanobacteria, such as the filamentous trichodesmium or the unicellular Crocosphera. But the more and more we sequence this NIFH gene, we come to find this, what we have eventually come to call non-cyanobacterial diazotrophs, which is a bunch of mostly gamma protobacteria in blue here, but it also has alphas and deltas and also some archaea. And these guys are much more widespread in the oceans. We can find them anywhere from the tropics to the polar oceans. We can find them anywhere from the surface to the mesopelagic. But we really don't know how much they are contributing to the global nitrogen fixation input. So this is what I'm trying to address here. So there's a fundamental difference when we think about these NCDs as compared to the cyanobacterial diazotrophs, and this is that these guys cannot do photosynthesis. So they don't have genes, they don't have photosystems genes, so they must obtain carbon and energy from some other source. And we suspect that this is an association with organic particles, but this has been quite an inferred, not really proved kind of association. So if you go to find these NIFH genes, either quantifying them through qPCR, for instance, you always find them in large size fractions, such as these 3 microns, 10 microns. You barely find them, very rarely in this whole fraction in the 0.2. Same with, in the right-hand side is the Terra Oceans data set, MAX, that had NIFH on them, same thing. So everything is like above 5 microns, but you barely find them in the dissolved fraction. But why will particles be a good niche for these guys? Well, particles are also thought to have an oxic core above a certain size and a certain density. This could be good for nitrogen fixation because the process is deactivated in the presence of oxygen. But also, as we know, particles are hotspots of organic carbon in the ocean, in a very diluted ocean. So if you take, for instance, this is a very simple experiment in the lab. If you take Vibrio diazotrophicus, a lab rat, so if you cultivate it without particles or without particles, it's only with particles present that they express nitrogenase. Nothing happens if particles are not there. So yeah, we have all this inferred information of that we always find these NCDs in supposedly particle attached fractions, but we have not really seen them or being able to measure how much nitrogen they are actually fixing. So this is what we are trying to do in my lab now, trying to go a little bit beyond into seeing them attached to particles, try to measure their nitrogen fixation rates. And eventually, and I think that we have talked about this in many of the breakout rooms, how do we want to extrapolate this very low throughput, if you're looking at single particles, how you're going to extrapolate this to a water mass or even to the global ocean. So first things first, how we're going to sample particles. You can do it in many ways. You can use sediment traps. You can use in situ pumps. We have used quite a bit marine snow catchers lately. So this is basically just a gigantic Niskin bottle of 100 liters, so you can deploy it to whatever depth you are interested in. You can allow it to sediment on deck for periods up to 5 hours or so, and that will separate particles qualitatively according to their sinking speed into suspended, slow sinking, and fast sinking. So last year we went on a cruise in the North Pacific, and we deployed this marine snow catcher at 150 meters across different stations, and we used that to do 59 incubations to measure both bulk and single cell nitrogen fixation rates, to do some imaging, and also to sequence an FHE. This is some of the preliminary data. So what you see here is in the size corresponds to the size of the particle, and the colors correspond to the nitrogen fixation rates. So what you clearly see at first sight is that nitrogen fixation happens in smaller particles, somewhere between 10 and 20 microns. And this was mostly in the suspended fraction where the smaller particles are. And this was kind of surprising because if we think about that, we need a minimum size of a particle to generate an anoxic core that will allow nitrogen fixation to happen. So this is something that we still don't have an answer to. We don't really understand how NCDs manage to get rid of oxygen in such a small particles to actually be active. The sequencing told us, once again, that gamma protobacteria were the major diazotrophs in these particles. So what we're doing now is trying to develop specific probes to some of the main taxa we found in our samples, so that we can spot them in the particle and eventually map that to nanosims analysis so that we can measure their isotopic enrichment. So in the right-hand side, this is a very fresh graph from last week. We pulled our stations together. We find that the higher nitrogen fixation rates occur in the fast sinking fraction, and the rates are somewhere between 1 and 2 femtomole nitrogen per cell and day. And just for reference, this is about the range of UCNA1 rates, which is a main unicellular cyanobacterial diazotroph in the ocean. So that means that NCDs could maybe, if they are abundant enough, they could be actually making a difference in how much nitrogen is coming into the ocean. So next is the problem of the extrapolation. So of course, nanosims is a very low throughput method. How are we going to upscale that to the full water column? Our approach for now, which is maybe a little bit naive, is to try to measure particle spectra using usual methods such as the LIST of the underwater vision profiler, so that we can know how many particles of each size range we have within a water column, so that we can extrapolate it just not by the volume of the water, but in a more coordinated way, so to say. But I assume that some people in the room have better ideas to do this with artificial intelligence or something more fancy. And last but not least, I'd like to also remind that it's not only NCDs that are associated to particles. So this is another data set from the South Pacific Ocean a couple of years ago. This is sediment trap data from anywhere from 170 to 1,000 meters. So we found that trichodesmin was actually the main diazotrophs in our traps. We did nanosims on those, and they were active. So to which extent are we considering particle-attached nitrogen fixation as just like a ways of exporting organic matter into the ocean, or is this also providing fresh amino acids for the deep ocean dwellers to do their thing, or to fix dark carbon fixation? So So yeah, these are questions that are unsolved and probably very patchy and depending on huge accumulations of biomass. And yep, so just some main take-home messages. So I think we need to explore a little bit more what the particles are made of, because it's not just size that matters here, but also the composition, the density matters a lot, I think, in what the biochemical gradients within the particle, and that will affect which organisms can attach to them or not. This will eventually help us resolve what is the role of NCDs in nitrogen fixation. And finally, I think that a big, huge problem is that how are we going to upscale this very low throughput data to the whole water column? And yeah, with that, I'd like to thank everybody in the NCDC crews and my lab and all the funding agencies. Thank you. Right on time.
Okay, bang on time, even a little bit early, so there's plenty of time for Q&A.
Sorry, Naomi Levine, University of Southern California. Awesome talk. Kind of to your point at the end, did you look at the carbon to nitrogen content of the particles by size, and whether the small particles were actually much more carbon?
By sinking fraction, not by size. Yeah, that's the main problem also with the nanozymes, that I will tell you the isotope ratio, but not the.
Yeah, because it seems like one of the kind of ways to understand when it's favorable for nitrogen fixation to occur, the C-to-N ratio.
Yeah,
probably.
definitely.
Yeah.
Cool. But this is more an analytical problem.
Yeah.
And what is the limit of detection of our equipment?
Yeah, totally.
Hello, I'm Coco. I'm a postdoctoral student of Yala Shaked. I work a lot with metagenomics, and one of the questions that I thought might be relevant for biogeoscapes is I ran into an issue where a lot of my mags actually contained NIF-H, but not any of the other NIF genes. And so I was wondering if you could comment a bit on whether this is an issue, or is it like a large percentage, or do you also see this occurring? Because, yeah, I can imagine for using NIF-H as a bioreporter, this could be an issue.
It is an issue. It happens very often. I want to say more in metatranscriptomes than in metagenomes, but I think that there are lots of pseudo NIF-H genes, so some things that are very similar to NIF-H, so that they are like misannotated as NIF-H, and I think that probably Bethany has more ideas on that than me. But yeah, it is an issue how we annotate it, and there's it's a paper from Tara, right? Using Tara data of these pseudo NIF-H genes, and I think there's a database that you can compare your metagenomes to rule that out. Can you?
Okay.
Ben.
Great, thanks. Nice talk, Ben Twining. Thinking about your challenge of scaling up to bulk nitrogen fixation, have you, and maybe I missed it, have you tried the sort of multiplying it by, your particle size distribution yet to see how close do you come to a?
We're on it. We're cleaning list data.
Okay.
I'll let you know.
I'd love to know. Yeah, we've struggled with it with iron. Thanks.
We're on it.
Okay, Maite here.
Yeah, if I saw it properly, but you seem to have activity at 1,000 meters for trichodesmium?
Mm-hmm.
Okay.
Yep. It was full of trichodesmium in the traps.
Okay.
Yeah.
And they are active?
And they were active.
Okay.
Thanks.
Last question, Rob.
Rob Medic-Nias. So if it's so far from the surface ocean and so far from a gas exchange, I'm not sure how big these processes are, but could you use maybe the deficit in nitrogen gas, similar like we use the apparent oxygen utilization, maybe an apparent nitrogen utilization to scale this up? Or is that just analytically not feasible?
Probably. I don't think we know how much that scales up to in the surface either to take it down to the deep ocean, but yeah, definitely ammonium production in the deep ocean needs something that will tell you that, right? But I assume that it's just taken up so quickly that it will be very hard to.
Yeah, but then the concentration changes. So if you measure the nitrogen gas, you can calculate what the nitrogen gas concentration in the water should be based on the temperature and salinity. So if you then measure less, you could attribute that to nitrogen fixation.
Probably.
Just an idea.
Thank you. No, I'm willing to try.
Okay, let's thank Mara again.
Thank you.